Hi, my name is Bernieve. I'm a senior. My mentor is Dr. Andre Wallace. So basically, my poster is about the role of INOS-1 in regulating cell migration during embryonic development in C. elegans. So just a background, we were very interested into that because cell migration is a very crucial process. It helps in maintaining multiple processes. So the gene that I focused on mostly is called INOS-1. So we were very interested in INOS-1 because from a previous research that Dr. Wallace did, they realized that INOS-1 is an enhancer for UNC40, which is one of the regulator for this pathway, which is the wave-scar pathway. So the wave-scar pathway is a very important pathway for cell migration in C. elegans. So the way the wave-scar pathway works is that those receptors receive signals from the cells, and then they transfer that signals to this GTPase, which gonna activate those components of the pathway. So the final result of the pathway is to get the formation of branch actin. So with branch actin, it's very important because it helps with the formation of the epidermal and cell in C. elegans. So for these pictures show you a regular embryos with no defect, no mutation. And the last three shows you if there's any defect, any mutation or whatsoever in the embryos. And this will cause the embryo to die if the epidermis is not formed. So we did a RNAi screen to get the INOS-1. So basically, from a frozen glycerol, we suspended using MPTG media, and from that we seeded the plates with about like 50 microliter of RNA. We let it dry for a few seconds. Then we added about like 10 worms that will be grown for about 5 to 7 days. Then we do either a screen for the embryonic lethality or for the change in brood size. So the brood size is different from embryonic lethality because the brood size tells you how the gene affects the phenotype of the worms. So from my results, you can see a regular SAX3 without the gene is about like 45%, but when it's added with the gene, it went down. And the same with VAB1 and UNC40. So the brood size also shows you the many differences between when those receptors are added with INOS-1. So also we're trying to find out the domain of INOS-1, and we realized that INOS-1 is an NAD binding domain, which is very important for signal transduction. So this is just like examples of where INOS-1 can be found. You can find it in humans, mice, yeast, and C. elegans, which is very important because if it was just in C. elegans, it probably wouldn't be as important for people to do research on. So finally, our conclusion, next day trying to find the exact location of INOS-1 into the wave-scar pathway. And also, by doing those research, they probably will understand and find out the exact function of INOS-1 into the wave-scar pathway.